Good day, this is the Professor, CBMD PhD, and today's Saturday, February 25th, 2023. It is 7:34 and 7 seconds, a.m. Eastern Standard Time, and today's research, telomeres. So let's hop to it here. The scientists make stunning discovery, find new protein activity in telomeres. Date February 20th, 2023. Source, University of North Carolina. As researchers made the stunning discovery that telomeres contain genetic information to produce two small proteins, one in which they found is elevated in some human cancer cells, as well as cells from patients suffering from telomere-related defects. As based on their research, they think simple blood tests for these proteins could provide a valuable screen for certain cancers and other human diseases, as these tests also can provide a measure of telomere health, because we know telomeres shorten with age. The telomeres contain a unique DNA sequence consisting of endless repeats of TTAGGG bases that somehow inhibit chromosomes from sticking to each other. As of 2 decades ago, the research laboratory showed at the end of a telomeres, DNA loops back on itself to form a tiny circle, thus hiding the end and blocking chromosome to chromosome fusions. When the cells divide, telomeres shorten, eventually becoming so short that the cell can no longer divide properly, leading to cell death. The scientists first identified telomeres about 80 years ago, and because of their monotonous sequence, the established dogma in the field held that telomeres could not encode for any proteins, let alone ones with potent biological function. In 2011, a group in Florida working on an inherited form of ALS reported that the culprit was an RNA molecule containing a 6-base repeat, which by novel mechanism could generate a series of toxic proteins consisting of 2 amino acids repeating one after the other. Researchers note in their paper a striking similarity of this RNA to the RNA generated from human telomeres, and they hypothesize that the same novel mechanism might be in play. They conducted experiments, as described in the paper, to show how telomere DNA can instruct the cell to produce signaling proteins they term valine arginine and glycine leucine. The signaling proteins are essentially chemicals that trigger a chain reaction of other proteins inside cells that then lead to a biological function important for health or disease. The researchers then chemically synthesize valine arginine and glycine leucine to examine their properties using powerful electron and confocal microscopes, along with state-of-the-art biological methods, revealing that the VR protein is present in elevated amounts in some human cancer cells, as well as cells from patients suffering from diseases resulting from defective telomeres. The researchers think it is possible that as we age, the amount of valine arginine and glycine leucine in our blood will steadily rise, potentially providing a new biomarker for biological age, as contrasted to chronological age. They think inflammation may also trigger the production of these proteins. When you go against current thinking, you are usually wrong, because you are bucking many people who've worked so diligently in their fields. Occasionally, scientists have failed to put observations from 2 very distant fields together, and that is what they did. As discovering that telomeres encode 2 novel signaling proteins will change our understanding of cancer, aging, and how cells communicate with other cells. Many questions remain to be answered, but the biggest priority now is developing a simple blood test for these proteins. This could inform us of our biological age and also provide warnings of issues such as cancer or inflammation. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.